From Microbe TV, this is Immune, episode number 34, recorded on August 24th, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
And from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here.
So Drew opened today, right? Classes?
Drew did open today with classes, just at the all virtual.
Yeah.
And, you know, it was the perfect day for there to be a worldwide Zoom outage.
Yep. Yep.
And what about Ithaca, Cornell, Cindy? Are they on yet or not yet?
They are testing and going crazy here. So, well, we're having move in. They extended over a longer period of time. Usually it's like 2 days, and it's 8 or 10 days. So they're all moving in. They are populating the local haunts, which they probably shouldn't be because most of them are supposed to be quarantining. Mm-hmm. But they test them as soon before they come onto campus. They test them right away. They get a 24-hour turnaround, and if they're positive, they're isolating them. And so the hope is that with testing undergraduates twice a week and testing faculty that have contact with undergraduates twice per week, that they will be.
Yeah, it's not bad. Twice a week isn't bad. That's pretty good.
Pretty good.
So, who's doing the testing on campus? Or?
The vet college where I am.
Very good.
They built a new facility. They got a test approved, everything. It's very interesting because if you're following all the different tests that are coming out, you know, clearly everyone's using the nasopharyngeal swab, but what they did was they did a head-to-head comparison, and they felt that the data showed that a front nasal swab was sufficient based on it's not as great as the nasopharyngeal at detecting the positives, but with a less invasive test, they're going to get compliance.
Sure.
From the students to be tested twice a week. So,
and it's a PCR test, right?
It's a PCR test.
Is it a regular PCR or LAMP or any of those rapid ones?
I don't know if it's the rapid one. I think it's just the standard qPCR.
Very good. Yeah. And then are they having in-person classes or is it online?
So there are mostly online, but they have guaranteed that each student, if they come to campus, would have an opportunity to have an in-person experience. So they're putting the pressure on the departments to make sure that at least some of the classes are in person. And clearly, you know, the science classes are going to be the main ones because they have laboratories associated with them and so forth.
Yeah.
So we'll see. But the class that I'm usually associated with, the essential immunology class, the undergraduate class, is going to be all online. And the veterinary class, which I lead, that's also all online.
Yeah, Columbia's all online, and they told students you can't come unless you don't have internet. They could accommodate certain numbers, right? But mostly people are staying at home. And we have a TWIV listener in Hawaii. His daughter goes to Barnard, right? And she said, I don't want to stay in Hawaii. I can't focus. So he emailed, he said, could you get her a place to live? I'm like, yeah, right. So I went around the elevators and I took pictures of all the roommate needed ads and I sent them to him because she wants to come to New York. Even if she's in her room taking courses, she'd rather do that than, I guess Hawaii is too distracting. Yeah, to get the beach out the window. Right? I'm going to go to the beach.
Well, it's also, you know, the 6 hour time difference, 9 hour time difference.
Yeah, that's true.
It's a little tough to take classes that way.
Yeah.
That would be really
I taught a summer virology course, and I had one student in Singapore, and I taught it at 10:30 a.m. and for her it was like the next day 7 p.m. or something like that. Wow. Weird stuff, right?
Yeah.
Anyway.
Yeah.
Well, Cornell, that's great. I'm glad to hear it because 2 months ago they didn't have testing plans and seemed weird.
Yeah.
And so I'm glad that they got their act together. That's really good.
I hope that I hope it staves off. I'm cautiously optimistic because I'm not sure that even if I were a college student now that I would follow all the rules. So I'm not sure that they will follow all the rules.
No, probably not.
It's good that there's frequent enough testing that if something happens, it will be caught.
Yeah. And, you know, they talked about having assigned seating too, so that they could easily trace back if a student was in the same classroom with another student who tests positive. I don't know if they're ensuring that happens or not. They're encouraging teachers, I think, to do that. And of course, the classrooms are like, they have to sit 6 feet apart, and not everybody can be there if they are in person. Sometimes there's A day, B day, or whatever. Yeah, it's an adventure.
Yes, absolutely.
Yes,
it's going to be very interesting. Should be very interesting in the next few weeks.
And I'll sit there and watch it all from my room.
Yeah. Very good.
It's both the best and worst semester for me to be on sabbatical.
Yeah.
I don't have to worry about any of the teaching things. There's some challenges in the lab, but I did not worry today about the worldwide Zoom outage.
Yeah.
Very good. Yeah, that's right. You're not teaching. Very good. All right. Speaking of adventures, we have a adventurous paper for you today. And it came out in Science. Selective and cross-reactive SARS-CoV-2 T-cell epitopes in unexposed humans. Big group of authors from La Jolla Institute of Immunology, Murdoch University, University of California, San Diego, and University of North Carolina School of Medicine. And first author is Jose Matus, and the last author is Daniela Weisskopf, but there's so many. Okay, we got these authors contributed equally. Spears, Sete, and Daniela Weisskopf, the last two authors. Mm-hmm. And one of the authors here is Shane Crotty, who will be on TWIV on Wednesday.
Awesome.
All right, so as the title says, this is looking to see if we're all infected with these common cold coronaviruses.
And there are a lot of them, right?
Well, there are 4.
This is where I can learn.
There are 4.
4?
Yeah, OC43, HKU1, NL63, and 229E. These will stay. You'll remember that for the rest of your life.
They have such descriptive names.
And do they all circulate all the time, or is it like flu where certain strains circulate?
They're always circulating, and over 90% of humans are seropositive for all of them. So we think they infect you in your first few years of life. You barely notice it. And then here's the cool part. Antibody immunity at least wanes within a year, and then you get reinfected over and over, but no disease or very, very mild disease.
Yep. They mentioned somewhere in here, because I had always thought of them as sort of all 4 being around all the time. And they mentioned 3 of them being more common than the other. Yeah. So they say that 229E is less common.
Yeah. So the first 2 of these were discovered in the 60s. Those were the first human coronaviruses discovered. And then after SARS in 2003, the next 2 were discovered because more people came in the field. They started looking more. So then we have 4. And so this paper is all about seeing if T-cell immunity is cross-reactive between them and SARS-CoV-2.
And was there known cross-reactivities between these other 4 or with the previous SARS that would have suggested this would be the case?
What kind of, you mean T-cell or antibody? I don't think.
Yes, both.
There's no antibody cross-reactivity. And nobody really looked at T-cell.
Yeah, I like, I guess, as we'll get into in the paper, the way that they describe how they started to look at this or why they decided to look at this in the first place. I thought that was a really kind of useful description of how they got to this idea.
Okay, I'm looking at this introduction here. 20, blah, blah, blah. Okay, anyway. You guys should walk us through this, don't you think?
Sure.
Because Cindy made lots of notes.
I did. I made a lot of notes because I wanted to make sure I understood it, and that's how that's how I always make sure I understand it. Yeah, I think before we start, I think the coolest thing is though that there's been a lot of anecdotes about people potentially having cross-reactive antibodies or cross-reactive T cells, and suspicion that maybe this would be protective. That's why I was asking you about cross-reactivity with other coronaviruses.
Yeah.
But nobody had really looked at it yet.
Right.
And so I think this is the first one. And I must say that when I first started reading it, I don't know about you, Brianne, but when I'm like, okay, they're culturing things for 14 days. and we can go over the details. But I said, how do they tell the difference between a memory T cell that's been generated against a different virus versus a naive T cell that gets activated in vitro?
Right. They actually, at one part, when they're setting up their experiments, they say this method is known to be robust for detecting low frequency T cell responses, including naive T cells.
Right.
And I was sort of like, did you just shoot yourself in the foot?
Well, yeah.
Well, I was hoping you would explain, because they say we ruled this out, and
right.
I don't know how they did it, but you guys.
I you know, we'll get into a little detail, but you know, I think from the upfront, it's important to say that I think the reviewers of the manuscript called them on that and said, no, you have to show us that they actually have markers of memory T cells, and that you can pull them out first and show that those can expand. And so I think, you know, by the time I got to the end of this paper, I was at least less skeptical and more convinced that they are actually looking at a memory T cell response. And this is important, right? Because if you think about it, you have a repertoire of T cells in every naive human, and within that repertoire, there should be T cells that can respond to, theoretically, pretty much anything, right?
Yes.
And so if you can expand them out, that doesn't tell you anything other than your person had a specificity of a T cell that could recognize SARS-2.
Got it.
And wow, really, that's not very surprising, right?
Yeah.
But the important thing is if they've already seen something, and they have a memory T cell sitting around that's already been activated, and that can be brought into play to fight against this new infection in a cross-reactive way. And so that's the fascinating part. But that's the part they had to prove to me, because otherwise I was like, I don't know, this is not exciting. So I don't know. What did you think?
So I thought a lot of the exact same stuff. So I guess for people who may not have as much background in this, a naive cell will have recently, or perhaps even not recently, been generated, but will not have seen antigen before. And so it will take a little bit of effort in order to activate that cell. That cell will take some time between when it first sees its antigen and when it's able to actually do something about that antigen.
Right.
It needs to both improve its function, it needs to divide, all sorts of things like that. A memory cell is going to have a much lower need for antigen. It's not going to need nearly as much stimulation in order to start to function. And that could be something that could be really helpful in the case of one of these infections. I always like to tell my students that they have to be very careful when they look at different types of data to determine if the data is showing, are the cells present or are the cells actually having a function? Right. And I usually make some joke about the fact that they might imagine sometime in their life where they have been present but have not actually been doing anything. They might be able to imagine something like that.
So let me ask you, if you take peripheral blood from people and you make PBMCs, how many would you need to throw peptides in to expect to see? I mean, would 1 million be enough?
Do you remember that? So Mark Jenkins has done some work on determining the initial frequency of the number of T cells with a certain specificity that are present. And I feel like it's in the 10 to 100 range in a mouse. And so I don't remember what it is in whether we can actually make an extrapolation to what it might be in a human. So you have very few of any one specificity,
right? But
there could be multiple different T cell receptors that could recognize the same peptide presented either in a different MHC or bound to a different MHC, the same, with different anchor residues, which we could go into if we wanted to. But basically, I think that I don't actually know the frequency with which it would be present in the PBMC population.
Right. And so your question is a little bit tricky, Vincent, because the answer varies a little bit based on what year you did your experiment. So
sensitivity.
So at the beginning, we sort of people had done some experiments where they would dilute cells and they could show that, yes, those cells were present at a very low frequency. But in a population of bulk PBMCs, like you mentioned, you couldn't always detect those low frequency cells.
I see. Okay.
Now we have more sensitive assays that will let you find those rare cells. It's called rare cell analysis. That will actually let you start to find them. And so my recollection when I was doing experiments with PBMCs was I usually was, working with around 1 million. I wasn't trying to do rare cell analysis then. So I'm not sure, but I think that it's sort of the same general setup.
All right. So let's dive in. So they have blood from people who have never been exposed to SARS-CoV-2, right? Samples from March 2015 through March 2018. So these people, no way they're going to be ever have seen SARS-CoV-2. All right. What do they do with their blood, Cindy?
Right. So they're going to take the T cells that they pull out. Well, the PBMCs are the peripheral blood mononuclear cells, and they have a bunch of different cell types in them. All the cells that you would find circulating in the blood, a predominance of T cells and B cells, but there's also antigen-presenting cells. And so what they want to do is they want to culture those with what they call megapools of peptides. And so what they do is they make pools of peptides from across the SARS-CoV-2 genome, including the spike protein, which everybody's really interested in, and the non-spike. And so they grouped it into these two pools, and they want to outgrow these T cells. Now, we already talked about, is this a memory T cell or a naive T cell? I think from this approach, they can't necessarily distinguish between those. Presumably, memory T cells would grow out faster and more robustly than a naive T cell, but you can't really discriminate that. And we'll talk about how they got it, discriminating it later. But they grow them for 14 days. So they incubate in pools of peptides there's antigen presenting cells there. They will get stimulated. They will stimulate the T cells, and the T cells proliferate.
So in total, they had picked 474 15 amino acid peptides, right?
That's right.
Yes.
I think it's funny that they say, spike and the rest. Like,
I know. For a T cell epitope, I'm not sure it matters that much, because when you think about all of the antibody studies that are done, that's kind of important because we're trying to look for neutralizing antibodies, right? And so they're almost exclusively going to be to the thing that sticks out the furthest from whatever it is you're looking at. And so in this case, the virus, the spike protein, because the spike protein is going to be binding to the receptor to infect the cells. So you want to look for things that bind to that. But the T cells doesn't really matter where the epitope comes from, because any infected cell should theoretically have every possible epitope made that's immunogenic. And so there's no real reason or rationale to divide these into spike versus non-spike.
The only thing that I thought about with that division is that, on TWIV, we've talked a little bit about how many of the vaccines are really focused on spike and have spike as their main antigen.
Yep.
And so this was sort of useful in determining how many of the epitopes that they're talking about might be found in one of those vaccines versus how many might be missed.
Right.
So I kind of liked being able to look at that myself.
Yeah.
And the distinction helped with that, but I think that's the only rationale I could imagine.
Okay.
Yeah. And the other thing that's important to note is you said 15 MERS, right? So they're 15 amino acids long. And that's also important because there's two MHC proteins. There's more than two. The two main ones, MHC class 1 and MHC class 2. And based on their structure and the way they bind to peptides, the MHC class 1 tends to bind shorter peptides of around 8 to 11 or 9 to 11 amino acids. And it doesn't really fit in these longer peptides. And so the longer peptides fit better into the MHC class 2. Now, that doesn't mean that a longer peptide couldn't be trimmed and bind to an MHC class 1. But generally, the 15 MERS are going to prefer an MHC class 2, which comes back to they found more CD4 T cells, which would be expanded by MHC class 2. So it kind of fits with that as well.
Yeah, so they don't really say much, or they don't see many CD8 T cells here.
Right.
That doesn't mean that CD8 T cells are not responding to this virus. It just means that they set up the experiment in a way that it was likely to detect CD4 T cells more so than CD8 T cells.
And our CD4 T cells are those helper T cells. And so when you're thinking about antibody responses, those are going to be the ones that are going to help with antibody responses. So, are they looking at the right T cells? Yes, if you're thinking about antibodies, they could have gone and done some CD8 T cell responses too, which may have been more informative as far as, protecting the host against infection.
Right. So I was just going to say the one thing that's important here with our question about memory versus naive cells is the way they set this experiment up is that they stimulated with the megapool, as Cindy mentioned, for 14 days, but then they stimulated the cells again.
Yes.
To look at their response. And so if these were memory cells, only that second stimulation really would have mattered. Only one stimulation would have been necessary. But because they do two, they could have activated some naive cells with that first stimulation and then further activated them with the second. And so that's why this experimental setup really doesn't let you distinguish between memory and naive.
And what they're measuring is the production of interferon gamma, right?
Yep.
So if there's a match between a T cell receptor and a peptide, the cell gets activated, makes interferon gamma, which they measure, right?
Yes, that's right.
Okay, got it.
And so because they do that second stimulation, what they did was use what they called mesopools. So just like something in between one or two peptides and a whole lot of peptides. But the megapools was basically just give it a large stimulation with lots of different things. So you get as many T cells to grow out as possible. And then using the mesopools with defined groups of peptides, they can then go back and deconvolute that to determine which ones, which T cell specificities there are. So I guess what they did was they had each peptide in multiple different mesopools. And so you could see which pools responded and use the computer to deconvolute and figure out which T cells there were actually responsive, or which peptides there were T cell responses to. And so what they did then was use flow cytometry to look at whether these were CD4 responses or CD8 responses. And so they were able to get 88 different responses, and 82 of them were CD4. So that's not a surprise because we said that the 15 MERS bind better to MHC class 2. And 4 were CD8, and then the other 2 were mixed. And so what's cool is that each donor, so each individual, naive individual, detected on average 11 of the 142 peptides. So that's not too bad, I thought. I thought that was a reasonable number. And so 54% of the peptides that they got T cell responses to were to spike, and even fewer to the receptor binding domain of spike. Which is interesting because that is what the majority of the vaccines are targeting for antibody responses. And this suggests that maybe we should include other epitopes, like you were saying, Brianne, to have both a T cell response and an antibody response.
Yeah. One part of that they also did that was really interesting is they looked at the number of peptides that responded. Compared to, or sort of relative to, the size of the protein.
Yeah.
And they showed that they had a lot of responses from spike, but that was also because spike was a really big protein.
Yep.
And so I thought that was sort of useful as well.
Yeah, because it would be interesting because if there was an enhancement so more peptides from a smaller protein, it might suggest that that particular protein is more immunogenic.
Yeah.
But if there's an equivalent, if you normalize for the size of the protein and you get roughly the same number of peptides, then there's no preference across the genome for the immunogenicity for the T cells.
Yeah, they say that the epitopes were evenly distributed across the genome in proportion to the size of each protein.
Yep.
And a lot of these other proteins are relatively small, like ORF6, 3A, and ORF8, et cetera. There are a whole bunch of them. So that's good.
Yep. And so one of the things you want to know is, okay, so what about the people who have actually had COVID and recovered? Do they have the same T cell specificities or are they different?
Yeah, this to me was sort of the place where I really liked their explanation of what they were doing and why. So they said that when this study was initiated, an assumption was that the in vitro T-cell culture epitope mapping would reveal an epitope repertoire associated with de novo generation of responses from naive T-cells. So basically, their goal in the experiments that they've already talked about, and that we've already talked about, was to make a list of T-cell epitopes that are seen in SARS-CoV-2.
Right.
And then, as they note, while these studies were in progress, we and others detected significant ex vivo reactivity against bulk pools. So there, they were looking at SARS-CoV-2 patients. They were doing one stimulation instead of the two stimulations that we mentioned. And they saw that there were responses in those patients. And that had made them start to think that T-cell responses were happening in response to this virus.
So the idea would be that those would be memory responses, right?
That's right.
That sort of could suggest that they were memory responses. Okay. And so they started this project to just make a list of T-cell responses. Maybe they were going to, who knows what their long-term plan was. And then while they were doing that, other data came out talking about T-cell responses in patients. And so they said, people are speculating that this might be due to cross-reactivity. And we have this nice set where we've just detected these epitopes, so we can sort of go ahead and test that. And I just really liked that paragraph where they explained that history here.
Yeah. And so they did. They found some T cells that overlapped with the COVID T cell responses, but then there were some where they were unique only to the people who had actually had COVID. And so it was interesting. So they say, for example, that they couldn't detect any T-cell responses from the naive people. They're not patients. The naive people to the M protein peptides, but yet that is a particular gene in which there are robust T-cell responses measured in people who have recovered from COVID. So that would suggest that they're not just looking at something non-specific, that once you get infected with the SARS-CoV-2, you do generate specific T-cell responses. But what they're saying is that in people who haven't seen it yet, there seem to be these T-cells that may be able to recognize some of the peptides from SARS-CoV-2 too.
Exactly.
So the next thing, clearly what they would want to know would be, okay, so you've got these, you can throw these peptides from SARS-CoV-2 onto these naive PBMC and you can see T-cell responses. But if the hypothesis is true that those responses are memory responses, they're clearly not memory responses to SARS-2 because they've never seen that. So they must be memory responses to something else. And that most likely thing would be another coronavirus that you've talked about, the 4 that you already mentioned, right? So what would you do next? Well, one of the things you might want to know is, of these peptides, what you're seeing responses to, how homologous are they to the analogous proteins from the other coronaviruses? If they're pretty analogous, that might explain how this is all happening. So they divided their 142 peptides into a group that never worked in their assay. So they never saw a good T-cell response to those peptides. Peptides in which at least one naive individual did respond, or peptides in which 2 or more people responded. And the idea here was that if more people responded, that's probably more likely an epitope that's going to have a very similar homologous peptide in another coronavirus. And so when they looked, the highest homology with the other coronaviruses was exactly that, in those peptides in which they had seen a response in more than 2 people. But nearly all, as you said, pretty much everybody gets a coronavirus in their lifetime once they're an adult. And so almost every naive person that they tested was seropositive for at least one other coronavirus. So that's good. So they were able to actually see peptides that have high homology and the ones with other coronaviruses. And the ones that had high homology were the ones where they saw more frequent responses, which would suggest that that's coming from another coronavirus. Did you have anything to add to that?
No, I think that was great.
Okay. So then what else can you do? This is where it got a little confusing to me, but I think I understood it. Then they took their 142 peptides and they binned them, not looking now at the T cell responses that they saw, but homology first. So you could look at homology first or T cell responses first, bin your people, and then look at the peptides that came out and see if they have homology. But the other way you could do it is you could take the peptides and look for ones with more homology or less homology, and then ask, are those the ones you saw T cell responses to? So you can look at it both ways. And so they took the 142 peptides and they said if they had greater than 67% homology with another coronavirus, and that just seems like an odd number to me, but they had some unusual calculation that they made based on length and this. I don't know. I think it was just
10
something they
amino
made
acids
up.
is enough.
I think they just made that. So I don't know. There's some logic there that I don't understand. But anyway, so 67% was their cutoff. And so if they had more than 67% homology with another coronavirus peptide or less than 67% homology with another coronavirus. And so they narrowed it down to 30 S peptides and 30 non-S peptides. And then they took and made specific pools from those and then asked, do they generate more or less of a T cell response? You agree with that?
I agree with that completely.
Okay. So when they looked at those, they found responses to those peptides. And what they found was that if they looked at the people who had COVID, one would expect they should have a good response to some of these. And they had a greater response to the SARS peptides than these homologous peptides from other coronaviruses. Now, that would be expected because they should have good memory T cells to SARS peptides. So they should have a very good response to the SARS peptides. They've probably also had other coronaviruses, but those other coronavirus peptides, maybe not as great of a response.
Because they're not actively infected with those. And
right.
so those T cells are not currently being stimulated in the same way.
Well, so they have memory to both, right? But they've more recently been
challenged
with COVID. So then when they look at the naive people, some of the T cells that they grew up against the SARS peptide actually recognized the other coronavirus peptides more strongly. And I think that this is the first evidence that would suggest that those T cells, presumably memory, that were in that naive person, were actually generated originally to that other coronavirus. And so that's the original peptide that they should respond to. And somehow that 67% homology, there's just enough in the way it binds to the MHC and interacts with the T cell receptor. There's just enough affinity that it can trigger those T cells well enough for the SARS peptide as well. Now, they also, of course, had some where they didn't respond at all and some where they were better to the SARS peptide because they originally put that in. And so it seems like there's a variety, a variegation, I think is the word they use, where there are some memory T cells that really were originally generated against another coronavirus and respond better to that other peptide, but they also respond to the SARS. And there are some that, for whatever reason, they just respond better to the SARS peptide or at least equally as well. But yeah, so I think that's pretty much what I got out of that.
Yeah, that I would agree. I think that, so here again, they're kind of getting a suggestion that, yes, this might be a memory T cell response, but they still haven't actually shown us that these are memory T cells. They haven't shown us that these T cells might not have some awesome reactivity.
Right. So then it comes to the question of how do you ask that? Like, how do you know if it's really a memory T cell? And what do immunologists do, Vincent, when they want to know if a T cell fits into a certain category?
Flow cytometry?
Yes. So they have specific markers that they associate with memory T cells. And, then of course it's not just, you can't just have one type of memory T cell. You have to have central memory, effector memory, TEMRA T cells, and so forth.
So in the last paper, Last Immune, we talked about a paper from John Ware's lab, where they were looking at all sorts of different types of memory T cells, and I thought I knew a lot about memory T cells since I did a PhD on them, and he had subsets I'd never heard of.
Yep.
So there are a lot of them.
There are a lot of them.
But we do have some markers that seem to be associated with memory T cells.
Right. And I actually liked this particular figure because there's a lot of data from a lot of different patients, and they're looking at different markers. So it's not just T cells against one peptide, there's T cells against all these different peptides. And it's pretty clear that the effector memory population is let me put it this way. The markers that are associated with effector memory T cells are the ones that are higher on those CD4 T cell populations that they grow out. And so the naives are really low, as one might expect. So it seems like these have, I would have put it this way, they have markers consistent with effector memory T cells. There's a little bit of central memory, they say. The data don't look that convincing to me on that.
And I will also say that if I were to imagine in my head the markers I would really be interested in for memory cells, the best possible markers, because sometimes people argue about markers, these are the ones I would pick.
Right.
So I also was particularly convinced because they picked some very obvious, good memory T cell markers.
And asking you here, so if you did a dual stimulation, so you stimulated once and stimulated again, would you expect to see those markers up in vitro?
I would expect to see the loss of CD45RA. I think the CCR7 is a little bit harder to say what I would expect in vitro. Right. But the CD45RA results I would expect. CCR7, not sure.
And I think it was a little bit later, but one other thing they did kind of to nail the hammer into the coffin on this, or beat the dead horse, or however you want to say it, was I think that the reviewer of the paper said, no, show me you can actually pull out T memory cells with specific markers and sort them into the naive population and the memory population based on these markers. Then do your expansion in vitro and show me that you're actually expanding the memory cell population. And they did do that.
Yes.
They showed that when they took those purified out populations and they put them with APCs and peptide, the naive ones didn't make any clones or grow out, and the memory ones did grow out. So that would suggest that at least under their culture conditions that they're really preferentially expanding memory cells with memory markers. And so it really reinforces the idea that these are in fact T cells that have previously responded to a different coronavirus that are present in a naive patient or person that theoretically, we don't know for sure, right? Theoretically can respond to peptides derived from SARS-CoV-2 should that person be exposed.
Exactly.
And what the function of that would be, we don't know, right?
Right. Yeah. So that's sort of where all of the debates and discussions start. And that's part of why this is so important and interesting. So the hypothesis or the sort of idea is perhaps because you have these memory T cells, that might influence your disease outcome when you are infected with SARS-CoV-2. So maybe if you had never been infected with a previous coronavirus, you would have a different outcome with SARS-CoV-2 infection because you didn't have these memory cells participating in a response. But if you had been previously infected, you'd have some memory cells that would influence your outcome. And that also might influence what could happen to a vaccine or sort of different patterns that we're seeing. And so that's kind of the next place where people take this, although there's some more evidence that we really need before we can make some of those arguments.
So the question that it comes back to is, would you be better off or worse off because the inflammatory pathology seems to be due to an overexuberant immune response, right? So is it better to have these T cells around or not? I would tend to speculate that having them around already, able to protect against the virus very early following exposure would be a better thing. I think the more that you allow the virus to replicate, the more robust of an immune response you need to generate, and thus the more likely you are to sort of get out of control and have immunopathology. What do you think?
Yeah, I would agree completely, but I think it would be very hard to do those experiments unless you're in some sort of animal model, because so many of us have been infected with these before.
Exactly.
Right.
And so it would be very hard to do a comparison and find some people who were never previously infected. And so, you could start to put in variables of how recently were you infected or which ones or things like that. So I think that it can be really hard to say without being able to do that comparison with these mythical people who've never been infected.
Yeah.
To really understand
the problem is that its memory, these T cell memory, lasts a long time.
Yep.
So essentially everybody has it. So I don't know how this could differentiate severe versus non-severe COVID-19,
Yeah, the only thing I could think of is if you had that, if those memory cells were generated more recently from a recent coronavirus, they might act somewhat differently.
They might bump faster.
But
also, you have to think about each individual has a different repertoire of MHC, which binds a different set of peptides.
Yeah.
And so if there are certain peptides that are more likely to generate a memory T cell response and other peptides that are less likely, there may be certain individuals with certain MHC haplotypes where they're more likely to generate a T cell memory response to another coronavirus that would be able to cross-react with the SARS.
Yeah.
But they didn't deconvolute any MHC information in these patients, so we don't really know. But given that they had at least, on average 11 peptides that they could respond to in each individual, it would suggest that there isn't really too much of an MHC restriction, although we don't really know the demographics of their population that they used.
Right.
I didn't look that carefully at it.
Yeah. I mean, the other big piece to all of this is related to that question of being present versus being able to do something.
Yeah.
So we know that these T cells are present in individuals. We don't know that they actually influence disease. We don't know, for example, that they could protect you from infection. We don't know. So there's this phenomenon of something called immunodominance, where some responses to some peptides are more effective than others. And we don't know if previous infection changes which peptides you respond to. Do you make more of the bad ones and fewer of the good ones, or we have no idea of sort of those details of how these T cells actually function to deal with virus. And so some people have taken these data and tried to say, oh my goodness, we all already have protective T cells in us.
Yes.
And there's nothing here that actually shows these T cells are protective.
Right.
They're present, but that doesn't mean they're doing anything.
Yeah,
that's right.
This was very widely publicized. There were people saying, we don't need 70% herd immunity. We're already there with these T cells. Unbelievable.
No.
Overinterpretation.
No. Yeah.
In fact, they say, the last sentence, they say, it's plausible to hypothesize that these T cells could be a contributing factor to variations, but this is highly speculative. Right?
Yes.
But then that part is left off in the press description.
Of course it is.
Yeah.
Not as sexy if you.
But how would you answer these questions?
Yeah.
It's hard, right?
I think it would be very hard to do. I can imagine some experiments, and I've seen some experiments like this done in animal models with some other viruses. So there are some experiments where people have done either infection or infection after, serial infections with different viruses, and shown that previous infection with one pathogen, there are actually some with bacteria too, will influence your outcome, and they can actually know the specific MHC types of the animal and look at the differences in which peptides are recognized and know which of those responses are more or less effective. But those, take a fair amount of knowledge of the immunology and MHC types and details of what's going on with that. And I certainly I'm not sure how you would do this in human populations.
And these are, as you mentioned, CD4 memory T cells, right?
Yep.
So at best, you could look at what cytokine production that are cytokines involved in B cell maturation and so forth.
Sure.
Right.
You can't do a CTL assay because that's a CD8, that could be really interesting to see if people differ, right?
Yep.
Yeah, I mean, it could be interesting to kind of do a study very similar to this, but instead of setting up the assay to be biased towards getting CD4s, you could set it up so that you're biased towards getting CD8s,
right?
And see if you see something similar or different with CD8s.
Yeah, because then if you could take someone never exposed to SARS-CoV-2, if you had CD8 T cells, maybe you could do a CTL assay and show that some people are better at killing infected cells than others. Who knows, right? That might give you some functional information.
Yep.
They have the last part, they talk about sequence homology. They look at the sequences of these epitopes, right? And they say, can we just look through the sequences and predict what might be a cross-reactive epitope? And they say 67% is a benchmark. What do you think about that? Are they all just based on sequence, or is there some structure involved?
So they're looking at primary amino acid sequence, and there's a lot to the peptide binding to MHC and the anchor residues, and different people having different MHCs that have different anchor residue requirements. And I think a rough 67% is going to be a tough cutoff, given all of the different things that go into binding.
Yeah, remember that the T cell binding is actually sort of making contacts with both the MHC and the peptide.
Yep.
And so details of MHC here are going to be pretty important too.
Yep.
Anyway, it's an interesting paper, right?
Oh,
yeah.
It's cool stuff.
So it makes me wonder how much, how much cross-reactivity normally exists to other things, right? Because when you think about a 9-mer or a 15-mer peptide, if the requirements are anchor residues, really the number of amino acids that contact a T cell receptor are not all 15, right? So once you start going down in numbers to, let's say, 2 anchor residues, and actually the things that contact the T cell receptor are maybe 2 or 3 amino acids, you're changing that the probability that another peptide has similar amino acids at exactly the same sites to make those contacts.
Yep.
And so it's not something we entertain frequently, but there could be actually more cross-reactive T cell epitopes than we recognize. And they looked specifically at other coronaviruses, which is about as close as you can possibly get. But there's nothing to say there might be a peptide in another bug, because there's like billions and billions of different bugs which might have a similar peptide. And so it's interesting, because if you think about how we study immunology, we typically use those basically completely naive mice that we grow in like super clean housing, and things don't always match up with what we see in humans, and we go, oh, okay, so mouse and human is different. But now that people are starting to use what are called outbred mice or pet shop mice, where they've had dirty parasites on dirty mice, where they've had parasites on board, or lots of different bacterial infections. Now, all of a sudden, the responses to new infections are kind of different. And they're different because of the shaping of the immune response, the repertoire, but it could also be some cross-reactivity that people haven't really looked at.
Yeah. So there's some really cool literature on that from Ray Welsh's lab, looking at LCMV and Pekinde virus that are not super closely related viruses, but they happen to have peptides that are close enough in terms of amino acids. And those papers really show quite nicely all the ways that your infection history influences your current immune responses to a pathogen. And so not only, do people vary based on their MHC types, but they also vary based on their infection history, which will be one of the many variables, I don't remember what they say in this paper, that influence what actually happens, a contributing factor to variation in what we actually see in disease state.
Mm-hmm.
Cool. All right. Thank you. That was great.
Yeah.
I wanted to summarize very briefly another paper which got a little bit of attention as well. Let me bring that up here. I've accidentally...
Called Longitudinal Analyses Reveal Immunological Misfiring in Severe COVID-19. So this one has also got a lot of authors. Are there multiple senior authors? I'm not sure.
They're not multiple senior authors, but there are 4 first authors.
Yeah, 4 first authors, and it arises from Akiko Iwasaki's lab at Yale.
Yep. And so Carolina Lucas, Patrick Wong, John Klein, and Diego Castro are the co-first authors.
So what I thought was interesting here is what they did a longitudinal study of patients. Right. That is, 113 patients admitted to hospital. They started sampling them right away and did so throughout their illness, taking a lot of blood samples from them. And, some of them got better and some of them got really serious disease. And so they said, can we see a difference in the immune profile, right? They looked at cells and cytokines. And the goal is to get a marker. So you can say, well, this person who just came in the hospital is having difficulty breathing. They're going to get really sick because their profile has this and this and this, right?
Right.
And other people are not. And maybe you give those people antivirals very early on and so forth. So, we're not there yet, but that's the goal. Mm-hmm. And so they find profiles. They have 3 different profiles that they say with various kinds of cells and cytokines that are sort of correlating with serious disease. They differ in the moderate disease patients versus the serious disease patients. They say we have identified a maladapted response associated with severe COVID-19 and poor clinical outcome, as well as early immune signatures that correlate with divergent disease trajectories, which means moderate versus severe disease. And there's a really good commentary by Stan Perlman where he summarizes it, but he also says why, we're not there yet. And the details are not so important, but here's what he says. It's really cool. He says, we want to have a profile. You come in the hospital, we do a profile of your immune cells and your cytokines. We say, you're going to get really sick or you're not. But he said, that's really hard to do on a patient-by-patient level because these markers vary so much, right? And so it's going to be really hard to get a number. And, the more markers you include, the harder it is to do that. And so he said, we really need to do a lot more. We're not there yet.
No.
In order to do that. He said, these kinds of markers are great for population-level analyses, but less for predicting outcomes in individual patients. Levels of specific cytokines vary substantially between people, making it hard to benchmark a level of cytokine expression that constitutes a sign of abnormality. Right. And they didn't mention that in the papers, and I think that's really important, right?
Mm-hmm. Yeah, it is.
So, and I don't know how you do that. I guess you have to look at more patients and...
Right. So statistics are great when you're looking at a population, right? So if you have 100 patients and they vary along this trajectory and you see these differences, it doesn't mean that the ones at the bottom are always going to do better or worse and the ones at the top are going to do better or worse. You can't really predict that, right? So the inter-individual variability is too high to be able to see where they are on any particular curve. We look at, large populations and then see the error bars are a little bit, the averages are a little bit different. And then we say these are statistically significantly different. But when you look at the populations, they're almost overlapping. So, yeah.
Yeah, I think the other thing that's sometimes really hard with this is that if you look at the reference numbers for what are the values in sort of a normal, healthy individual, those reference numbers are quite diverse, and they're not something that people spend a ton of time studying. People are not usually really excited to know how much IL-2 is in people on Tuesdays.
Yes, exactly.
Healthy people on Tuesdays. And we actually need those types of numbers so that we could make these comparisons. And that hasn't been a big priority.
Yeah. He said we need to get more samples from people with very early signs of disease before they go to the hospital, right? Because maybe by then it's too late. And then compare those to people that never go to the hospital. I think that's really important, but that's hard to do because especially the very mild infections, you never know where they are because they're not close to a hospital, right? And maybe
you
Cornell.
need to hook up. I was just going to say, you need to hook up with the universities where they're
it's true.
testing these individuals repeatedly.
Yeah.
And as soon as they test positive, regardless of whether they are having any symptoms or not, then you should start a study with them.
In fact, going back to school is a great opportunity to do this because you're going to have people who are positive, but they feel fine. So
hopefully someone does that. Please go do that.
Well, Yale has a college, and its people are in part from Yale, so they could do it. Now, the one thing that I really was interested in here, they have, of course, they take nasopharyngeal swabs of these patients and they measure viral RNA over time.
Right. Yes.
And what's very interesting is that the people who develop mild disease or moderate disease, their viral loads go down, right? But the really sick people, they're maintaining their viral load, right? And so Perlman speculates that maybe, they have a poor initial response to infection and it gets out of control and it just persists longer because of that. And the host then is over-responding because, he says that it's clear that in these severely ill people, it's an immune dysfunction of some kind. And maybe it's triggered because you don't have a good early response. And so, thinking of the previous paper, maybe if you have some cross-reactive T cells, they can control infection very early on and others don't, right? So I thought that was really interesting.
The other thing I thought was interesting is they talked about these cytokines that one normally wouldn't anticipate to be associated with a viral infection.
Yes,
so,
right.
IL-17, the Th17 type of cytokines, as well as Th2 type cytokines like IL-5 seem to be elevated, which is interesting because it makes you wonder whether the immune system is confused and starts to generate the wrong type of response, because we like to have the immune response paired to the type of pathogen so that you're generating the right type of response at the right location at the right time.
Is that where misfiring in the title comes from, you think?
Maybe. I don't know.
I think so.
Yeah, we can't guess, but it's an interesting use of the words. Anyway, so that's cool. I think that's very interesting and more to come.
The other thing that's really neat is that they're all lymphopenic. So they all lose CD4 and CD8 T cells. One thing interesting to know is, do they lose the types of CD4 and CD8 T cells, or the repertoire of CD4 and CD8 T cells? Are they differentially affected in the healthy versus the ill? I don't know. Maybe they lose more T regulatory cells in the more severely infected patients. They have a less controlled immune response.
Yeah, I think there's a lot of really interesting stuff here, and it just also highlights a lot more cool experiments one could do.
Oh yeah, definitely.
Yeah, I hope they get done because, SARS-1 probably would have been equally interesting, but it stopped after 8,000 infections. And so, not much
thank goodness,
done.
Because the fatality rate was much higher.
Yeah, I don't want it to keep going, but I'm saying this one is the reason it's getting all this attention, because it's continuing and so many millions of people are infected. I just hope that if we get vaccines and stop this, that the research doesn't stop, because it's fascinating, right? There's a lot of aspects that can be many, many things that follow from this. So
I think that there's a real demonstration of the power of some of these big data, multi-parameter human immunology studies.
Yeah.
Yep. I think it's been really fascinating. They've been there and people have been publishing on this stuff, but now I think people are starting to say, whoa, you really can learn a lot from this. Yeah. It's not all descriptive.
Yeah. And you know what? What I think is really interesting, many immunologists that never thought about viruses are now working on this.
Yes, exactly.
It's amazing. And these people who I said, the immune system is there for pathogens, right? Why don't you think it? And then, they're developmental immunologists and all this. And now they're gangbusters. So good. That's good.
We're going to learn a lot.
As the one person in my immunology graduate class who cared that there was a pathogen, everyone else was really interested in autoimmunity and things like that. I feel very vindicated.
Yeah, it's just great. It's just great. Okay, let's. We got a big mailbag. Let's tackle some.
Oh, we do. Yeah.
This first one is from Matt, who writes, I sure enjoy your fascinating immune podcast. I have not heard of any mucosal delivery vaccine candidates for COVID-19 since this virus does so much of its damage in the lungs. I was wondering if such a route might be helpful, such as intranasally. And Matt sends a review article, which is a really nice summary of different ways you can develop mucosal vaccines of different sorts. And he asks, what are your thoughts? Well, we just covered in TWIV a adenovirus vectored vaccine out of China where
we did.
They not only injected it intramuscularly, but they instilled it in the nose. Was that what was the model, Brianne? I forgot. Was it non-human primates?
Yes, it was. It was rhesus.
And the interesting thing is when you do an intranasal.
No, actually it wasn't. It was mice because I remember thinking about how hard it would be to do something intranasal in a mouse.
Oh, they just they use a pipette tip.
We do it.
No, stick it there and they sniff it in.
Yeah, I know. I've done it with guinea pigs, and I just remember how much they
it's harder with mice.
It out.
Yeah.
You have to kind of lay them on their side and make sure it stays in, right?
Yeah.
Not drips out. And when you do intranasal installation of the vaccine, you get protection of both the upper and lower tract. Mm-hmm. Whereas if you do IM, you just get lower tract, which is great because you prevent disease, but you don't necessarily prevent transmission. So,
right. They saw a different immune response between the two vaccine deliveries as well.
Yes. And I have to say, Amy called me out. He said, you didn't talk about the different cytokine responses. That's what's really important. Yes, absolutely right. So I agree. And I'm glad to see that one, because I do think some kind of mucosal immunization, even gastrointestinal immunization. I think there's an adenovirus.
I think there's several candidates that I found. So they're out there. There's a couple companies that are working on the mucosal vaccine for.
Yeah. So there are, and I think they're important. I agree.
What's the latest count? Do you know how many vaccine candidates are in the trials right now? It was 140 something before, wasn't it?
Oh, it's even more.
150.
I will get it. I have the thing here. But so Brianne, you read the next one, please, and I'll get the answer to that.
All right. Robin writes, I sent my first email question included below to TWIV, but then I found and listened to your immune podcast, COVID-19 Part 3. You mentioned that many people do not seroconvert, meaning that their body does not recognize the COVID virus as a threat and thus does not take the steps to fight it. This was one of the questions I had, but then I want to know if you think such non-seroconverters will continue to carry this virus silently their entire rest of life. Are there other infectious viruses that some folks carry silently? It seems the more we find out, the more questions arise. Robin from San Pablo. And Robin is a physical science teacher with a life science education and just enough knowledge about the subject of microbiology to possibly be dangerous.
Mm-hmm. The second part of that's easier, right? So
yes.
Are there other infectious viruses that some folks carry silently? Absolutely.
Yeah.
Because they're DNA viruses specifically.
Yeah.
Yes.
Like the herpes viruses
viruses, chickenpox virus.
Yeah.
And there are some like anellovirus is like one called Torqueteño virus. I know Vincent likes to say
That's
that one.
That's a good one. I do. It's anello because it's a racanello virus, anything that sounds like me, I like.
It's
Especially
in
if it's a virus.
Lots of people. It's over 90% of people, and does not seem to cause any disease. But most people silently carry that virus.
Yeah,
a lot of people carry CMV, EBV, Epstein-Barr virus.
Yep, herpes simplex.
A lot of the herpes simplex,
HHV 6 and 7.
Yep.
And then a lot of polyoma viruses are silent unless you're immunosuppressed. Yeah, it's just, we're full of them.
But the question is that he wants to know is could non-seroconverters carry SARS-CoV-2 silently the rest of their lives? Now, is there evidence that coronaviruses persist?
I don't know of any.
And I don't know.
I think the evidence, I don't know how long ago we said this, but now there have been many serology studies, and I think most people are seroconverting after infection. Would you agree?
I would agree. I think that people should remember that seroconvert means having a positive antibody response that can be detected. And so some people don't seroconvert, but that doesn't mean they're not making any immune response. They could be making a T cell response here, or their antibody levels could wane and could go down to a level that's harder to detect. And so I think that there are some people out there who are antibody negative for one reason or another. They are in the minority, but I don't think that means that they're not making any immune response. It just means that we're not detecting their antibodies when we do the measurement.
Yeah, I just read a paper where they looked at serological studies, and they say, contrary to what's been said, we think basically everyone makes some kind of antibody response. And I think the early ones were just the technology was being worked out and so forth. And, you get infected, you make some kind of immune response, right?
Right.
All right. So, Cindy, how many vaccines are there? 203 in development.
Whoa.
And the Milken
and one will win.
Hopefully more than one, because, we need many billions of doses, right? To hit everyone. And some 1, 2, 3, 4, 5, 6 are in phase 3.
Oh, wow. That's
and
great.
1, 2, 3, 4 are in phase 2. So that's good. And the rest are either in phase 1 or preclinical development. Yeah. So 26 vaccines are in clinical testing, which means phase 1, 2, or 3. Okay. All right, Cindy, can you take the next one?
Sure. David writes, "Hi, immune hosts. I have been listening for the last 10 to 15 episodes, which I started a year ago or so, to learn some immunology, since my field of cancer biomarkers has made a sharp turn in that direction. However, now with COVID-19, I am reading and learning some virology. My last formal class in virology was in med school over 30 years ago. I now run an anatomic pathology lab, and I am boarded in anatomic pathology, but not in clinical pathology, where they study and perform serologic testing. Since I use a lot of antibodies in my tissue biomarker work, I know how challenging it can be to get specific antibodies. I'm not too surprised at the lack of respect we are seeing for serological testing for spike protein or other viral proteins. I am more surprised about the concern that the antibodies may not have neutralizing activity. I understand that cultured cells test for neutralizing activity, but if most of the antibodies generated by infection did not have neutralizing activity, with over 1.5 million cases in the US, wouldn't we see hundreds of cases where healthcare workers got reinfected and became symptomatic? I have not seen a single case report of recurrence that is both sufficiently spaced, say test positive after greater than 30 days with negative tests in between, and symptomatic, say requiring hospitalization. Are there cases out there and not reported? Or if not, isn't this pretty compelling evidence that the vast majority of infected individuals making neutralizing antibody, which could be detected by serology? Thanks for considering this question. Sincerely, David. And David is at Yale. So if you asked us this 7 hours ago, we would say no, but your email came on just the right day. And apparently there is a brand new study from Hong Kong, right? Yeah. That suggests that there is an individual that for sure was reinfected because they were able to sequence the first time and sequence the second time. Is that correct?
Yep.
And it's not the same virus. It's definitely not the same virus.
That's the key, to get the genome of both, and then you can really show that it's a different virus. Yeah. But the key is that person did not have symptoms.
Get sick.
He didn't get sick.
The second time.
Yeah,
or the first time.
which is what you see with these, as we said, with these seasonal coronaviruses. That's the pattern that you see. But, I agree with the logic, though. We're not seeing a lot of well, it's still too soon after the initial infections. But, as we've said so many times here, T cells clearly play a role, too, and we generally don't measure those.
Right. Antibodies are not the whole immune response.
And they're easy. They're easy to measure, right? Antibodies are pretty easy.
They're easy to measure. Yeah. So there's been, but this is not the first story of this, though, right? There's been a number of anecdotal descriptions of reinfection, and people have said, no, that's not true. We don't believe it. There's not enough evidence, and so everybody's been saying, no, you can't be reinfected, and now.
But that's like one out of how many millions?
A lot.
But the thing is, the previous ones, there were just PCR, and it was, you didn't know if it was the same virus or not. It could just be that person, we know they shed bits of RNA for long periods. But if this is true, and I haven't read the paper yet.
I haven't read the paper.
I'm not surprised. And, the thing is, he didn't get sick, and that's the key.
Right. That's the key.
So some people are starting to say this, means terrible things about vaccines.
No, it doesn't.
But it doesn't at all. So some people will look at it and say, oh, well, that means that the immune response can't be protective. And so you can get reinfected.
No.
The immune response that you make with a vaccine isn't necessarily exactly the same as what you make with natural infection. The virus, for example, could be inhibiting part of the immune response, and the vaccine perhaps doesn't have that inhibitor, and so it lets you make a really great immune response. And so just because reinfection happens with natural infection, that does not mean that it's not possible to make a protective immune response.
Right. And the other thing is people think, they try and equate having a vaccine with sterilizing immunity, so that your body will never, ever be infected again. And that's simply not the case, right? So it's not that, if you get exposed again, that no virus, not one single particle will ever be able to infect a cell. That's not true. It's not possible, right? So there's going to be a little infection, but you have enough memory, because the memory was a big part of what we've been talking about today, around the memory B cells making antibodies, memory T cells around, that you nip it in the bud really fast. You don't feel sick, and most importantly, don't shed a lot of it, right?
Yeah. So those memory T cells and memory B cells are activated more easily,
right?
And don't require as much time, just like we were saying before. And so you get reinfected, they act immediately and get rid of that virus before you even knew it was there.
Right. And that's the reason why we want these vaccines. That's the reason why you want a vaccine to begin with, is because it's more dangerous to have that primary infection, because something could go terribly wrong. But if you have a vaccine, it's much safer. And then you have the memory responses around to protect you the second time you're exposed.
Of course, once everyone's infected or vaccinated, then it doesn't matter anymore. And then young kids who are born and get infected with SARS-CoV-2 don't get terribly sick. And it's going to be like another seasonal coronavirus. It'll be the 5th seasonal coronavirus. And I would predict that we're not even going to use the vaccines in 5 or 10 years that are all being developed now. Mm-hmm. But we'll see. I think the vaccine could be different, as Brianne says, but it could also be the same and allows reinfection in the absence of disease. Who knows? We'll have to just see. And it's going to take more than 3 or 4 months. We have to wait a year or two to do long-term studies, right?
Yep.
It's a really interesting situation.
But you shouldn't think of it as a catastrophe.
It's not a catastrophe. Absolutely not. But of course, that's the way the press is playing it. Oh my gosh, immunity doesn't work for this virus. No, it does.
It's exactly what you want it to do.
Brianne, next one, please.
All right. Mei Yi writes, hi, I am a Bi 115 virology student from Caltech. In Immune 29, Immunology of COVID-19, you guys mentioned that IL-6, IL-1, and IL-8 TNF were upregulated in patients with COVID-19. I was wondering if these proteins are specific to the response to COVID-19, or if that's just part of a general inflammatory response. Is there a protein or group of proteins that are specific to COVID-19 and would help us identify that inflammation is caused by SARS-CoV-2? Thank you, Mei Yi. You are exactly correct that these are sort of the classic proteins that are in the general inflammatory response. Mm-hmm. So that's what we're seeing here with these. And we would hope that maybe something like the paper we talked about today from Iwasaki's lab could give us a combination. It's probably not going to be one protein, but a combination of proteins that are specific, but we don't have that yet.
Nope. And the previous paper that I talked about in the last immune, we talked about a bunch of different markers. They used 40 or 50 different markers to look at surface markers and cytokines they're making and different effector molecules that they can make. And so people are trying to figure this out. Would it be specific for just SARS-CoV-2? I don't know. I mean, probably maybe RNA viruses because they replicate in similar ways. You'd want a similar immune response to those. I don't know that you'd need a exquisitely tailored immune response to any particular organism as far as the cytokines that you would make. The T cell and the B cell receptors, the T cell receptor recognizes the MHC peptide complexes and B cells making specific antibodies. That's where that specificity comes in. But there should be general signatures of different types of infections.
Let's do one more round, okay?
Sure.
Sarah, right?
My first? Okay, you're first. Go.
My name is Sarah, a medicine resident training in a New York City hospital. We've been at the forefront of the COVID epidemic. Actually, I think it was your turn. I'm sorry.
No, go ahead. That's fine.
It's been surreal, but thankfully at this time we are trying to get back to normal. But who knows how long that will last. I have really appreciated listening to Microbe TV. I have definitely learned a lot from you all. I've been curious about how SARS-CoV-2 is able to do asymptomatic shedding. How is the virus able to replicate easily while the host does not have symptoms? Correct me if I'm wrong, but I assume that symptoms occur once the immune system is alerted. So going with that reasoning, I'm assuming that the host is able to basically replicate with an invisibility cloak for many days. I think you mean virus is able to replicate.
Yeah.
Do you have any insight into this? I know that other viruses like flu and herpes do have abilities to hide from the immune system. So wondering whether you could also extrapolate from other examples. I would really appreciate some answers, as I have been looking all over the internet and could not find anything. Well, a lot of people are confused by this. How can a virus reproduce without causing symptoms? Well, as we just mentioned, for so many herpes viruses and polyoma viruses and others, they do. And it's not that your immune system is not responding. It is, because if you measured cytokines in an asymptomatic person, you would see some difference, right? Mm-hmm. But you don't feel anything. And why is a great question. It may be that you have to be under a certain level of titer, but I'm not aware of a lot of studies of asymptomatic people, but they seem to have equal viral loads as far as SARS-CoV-2 goes. So why you don't have symptoms, why you don't feel fever and ache and so forth, I don't have an answer to that. It's a good question. You're not going to find it on the internet because there is no answer. Right? What do you guys think?
But there's a difference between asymptomatic shedding and pre-symptomatic shedding, right?
Sure.
So because somebody could be pre-symptomatic and eventually they're going to feel sick, but they just don't feel sick yet, even though they tested positive.
Yeah.
So I think, it might also depend on, like we were saying in the second paper that we're talking about, the timing, right? The kinetics of the experiment. But, I also think that different people have different pain tolerances.
Yeah, for sure.
Yeah. And they probably have different fever tolerances.
In fact, there was a paper.
Somebody could probably run 102 fever and not feel that bad.
There was a paper we did on Twitter weeks ago about a study in China which said, 20, 40% asymptomatic. And they did a survey and the people, they would ask the people, how'd you feel? I'm fine. And they said in the discussion, fine to one person is not fine to another person. So it's absolutely right. So
some people are much more in tune with their body, right? And some people, power through life and totally ignore what's going on with their body. They have pretty serious things going on and ignore those symptoms.
Yeah.
Right?
You could ask some people how you feel. I'm great. I have no problems. And other people, I feel headache, muscle ache.
Well, or you could ask, me some days, how I'm doing because you want to know about COVID, SARS-CoV-2, and I could say, oh, I'm fine. When really have a pretty stuffy nose, but I'm just assuming it's allergies.
Right.
Yes, exactly. That's another way. Thing. That's another thing. Yeah, assuming it's something else. Right. So it's a tough one to sort out, but very interesting.
And there are a lot of things on the internet now that you'll find where they're trying to say, is it allergies, is it flu, or is it COVID, right? But still, even so, there's a lot of overlap.
Yeah.
So you just don't know. So is it me?
Yes. Go ahead.
Okay. So Sophia is the last one. Yes, Sophia. Hello, beautiful immune team. I hope you are well. I saw this in the news, and so there's a link to a Science Mag article. I'm not sure if the above is worth talking about, but my question is, if we are concerned about vaccine enhancement with this virus, is the above study a good idea? Now, the study is on the tuberculosis vaccine, right?
Yeah.
Yes.
And so, or is vaccine enhancement dealing with another arm of the immune response? Any ideas? Maybe a naive question. All the best from Greece. 9 degrees C, windy and rainy, great quarantine weather. Following to my previous email, here is another study that does shed light and does explain the death rates in different countries through the BCG lens. And so there's a medRxiv paper here. And says, in essence, countries with robust BCG vaccination programs since 1922 seem to have protected their elderly population somehow. Does it still work? Well, it looks like through this vaccine. If this is the case, I have hopes for Africa. P.S. Great immune episode last time. Greetings. And so I think I did mention this once before, the study on this BCG, because it's kind of interesting. So the idea here is people have been using BCG. So Bacillus calmette-guerin. I can never say it correctly, but yeah. So BCG is a live attenuated vaccine that they use against tuberculosis. Now, it isn't a particularly great vaccine against tuberculosis. People still get infected. They still have serious nodules in their lungs and so forth, but they seem not to shed as much or get as sick.
It's kind of protective in kids,
a little bit,
but not as much in adults.
Right. So a lot of countries have stopped using BCG, and so they can track when countries have been using BCG and when they haven't, and they can track the COVID attack rates and so forth. And my understanding was that they could explain some of this away. So the initial study suggested that the countries in which they had they regularly used BCG versus not were somewhat more protected. But then when you looked at the demographics of the population, it was a little bit different. And so I don't know if that was all based on symptomatic disease. And so it could be that there are younger populations in the ones where they're still using BCG, and in the older countries, the countries where they're not, there are older populations that are more susceptible. So I'm not sure that it's a really great study yet on this.
Yeah. So there were a couple of different hypotheses that were discussed about this. One, that BCG had some sort of general population effect, or one that getting BCG sort of around the same time as you were infected might sort of give you a bit of a boost to your immune system through this type of immunity called trained immunity.
Yep.
There's a person named Mihai Netea who does a lot of work on that. And he actually had a paper that came out recently in Cell Reports that looked at some data on the trained immunity hypothesis. The paper is called Safety and COVID-19 Symptoms in Individuals Recently Vaccinated with BCG, a retrospective cohort study. And this is one of the highlights from the paper. They have little bullet points of highlights. BCG might be associated with reduced incidence of sickness and extreme fatigue. So they do have a pretty nice study that just came out within the past month, but the effect is subtle.
Yeah. That would not necessarily surprise me.
Right.
So there were some studies when I was a postdoc on CpG DNA triggering TLR9, or well, we didn't quite know that yet at that point, but that it could boost innate immunity in a not very non-specific way and give you a slight protection against later challenge. And they did things like challenge with Ebola, and those who had no specific vaccine, just a TLR agonist, seemed to be somewhat better protected. So there seems to be something about boosting your innate immune response that might be protective. So that's not such a surprise. But once you've had this vaccine for years and years and years, it doesn't make immunologic sense, right?
Yeah, that doesn't make any sense to me.
Unless there's a lot of cross-reactive T cells or B cells. But it seems like the specificity wouldn't be there.
Right, exactly.
There's a few clinical trials ongoing to test this in a rigorous way. So
the polio trial I know you've talked about before.
Yeah, other virus vaccines too. So we'll know soon enough, I guess. All right.
And Sophia wrote something else. Do we want to just mention that quickly?
Yeah. Of
because
course.
she said we don't do picks of the week anymore, but that's only because we talk too much. but we should do some. But says when you go back to doing this, here's a nice story coloring book on immune defenses on viruses. And I opened that up. I don't know if you looked at it. It was really neat.
It's very nice.
It was really cool. So although I'm not sure what the cytotoxic gun is about, but for a more intellectual and complete version, I recommend TWIV 597, an outstanding and most enlightening episode. All the best, Sophia. So thanks, Sophia. We can put that in our links so people with children can have their children color in the coloring books, or if you so choose, you could yourself if you wanted to.
It's a cool book. It's nice.
It was really neat. I like that.
It's very nice. I hadn't seen that. It's from Arizona State, I think, and a couple of other places. Cool. Thank you, Sophia. All right, that's Immune 34. You can find it at microbe.tv/immune on any podcast player. Please subscribe so you get every episode. And if you have questions or comments, send them to immune@microbe.tv. If you like what we do, consider supporting us. You could become a patron, like a patron of the arts, the Medici, right? Well, you don't need to give us millions of dollars, but you could give us a buck a month if you want. Of course, if you want to give us $1 million, you can do that. That's fine. We'd be happy to take it and use it wisely. Microbe.tv/contribute. Cindy Leifer is up at Cornell University on Twitter. Cindy Leifer. Thanks, Cindy.
Thank you.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. And speaking of Neil, hopefully next time we'll have Steph back to join us.
Yep.
Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.